and explain to the auditor about the method validation and sample analysis workflow in our laboratory during the recent audit in April 2021. He was a assistant manager in Power Science, Chennai, Tamil Nadu, during the period of March 2017 to Feb 19, and he worked as a project in charge for the method development, method validation, and subject sample in the biological sample using LCMS. And he developed the methods of molecules such as penicillamine and bupropion, isosorbide dinitrate, and fitonite, etc. And he was a scientist in Power Science, Chennai, Tamil Nadu, during the period of Jan 2015 to Feb 2017, worked as an instrument chemist and involved in the method validation of LCMS and MS methods. And he maintained a routine maintenance and calibration of the analytical instruments. He worked as a scientist to Navitas Life Science, Manipal, Karnataka during the period of Jan 12 to 2012 to Jan 2015. He worked as a sample processing chemist and operation of thermo LCMS in the method development and method validation. He was also a bioanalytical executive at Bombay Bioresearch Center, Mumbai, Maharashtra during the period of Feb 2011 to December 2011. He was a trainee in Astes, Chennai, Tamil Nadu in the August 2010 to Feb 2011. So I extend my heartfelt thanks and welcoming you for the session, sir. Session is over to you, sir. Thank you.
Thank you so much, Amalraj and Nonaja, for the brief introduction. Am I audible?
Yes,
sir.
Chandru sir, you're highly audible. And before the sessions, I would like to introduce our participants to you. Here we have faculty members from diversified field and from all over the India. Faculty from biotechnology, faculty from biomedical engineering, faculty from computer science background as well. They are connected here. It is for your support and your information, sir.
Okay.
And thank you for your time, valuable time you are sharing with us. And session is yours, sir. Thank you.
Yeah, thank you. I'll just share my screen.
Yes, sir.
Is my screen visible? Is my screen visible?
No, sir. We having visible screen. I mean, share the full. If you share the full screen, it would be fine, sir.
1 second. Okay.
Yeah, perfect.
Yeah.
We can see your screen.
Yes. Okay.
And sir, we have 1 hour session and followed by questionnaire. So if you could concise your talk in an hour, it would be fine, sir.
Yes. Sure, Amalraj.
Thank you.
Okay. Thank you all so much for your presence for this session. So basically, my topic will be around clinical sample analysis. As we have seen, most of my experience is in the analysis of sample. Basically, in bioanalytical department, I am working in bioanalytical department. In this department, we will receive samples from clinical site. So our aim is to quantify the amount of drug present in each sample. So I will talk about the sample analysis part in detail in the coming slides. So why is this clinical sample analysis is important in research? See, without analyzing the sample, we cannot tell whether this drug has reached the site of action or not. So this analysis plays a very important role in determining the effect of drug in our body. So these clinical samples are essential for developing new treatments and understanding the disease state. So they provide data that researchers rely on to make informed decisions about patient care. By analyzing these samples, scientists can identify biomarkers, assess drug effectiveness, and improve patient outcomes. So this is the drug development process. As you all know, initially it's starts with discovery. There we identify the drug targets and candidates. So once we identify this, once the discovery part is done, before conducting the trials in humans, we conduct preclinical studies. Means before testing in clinical phase, we have preclinical studies. In this preclinical studies, we will evaluate the safety and efficacy of drug in vitro, which means in lab, and then followed by we will check in animals. So here also we will quantify the amount of drug present in animals in preclinical studies. Then once the results are satisfactory in preclinical studies, once this safety and efficacy is evaluated in preclinical studies, then it will move on to the subsequent phases. Like phase 1, it will be done in very limited healthy volunteers with a limited group of population to assess the safety and dosings in human. Then later on, with some increased population, it will be evaluated in phase 2, subsequently in phase 3. Once phase 3 is completed, then it will go for regulatory review based upon all the data collected. So from each phase, we will get samples. We will receive samples from clinical site to bioanalytical site. In bioanalytical site, we will determine the concentrations present in each sample. Based on this data and other related data like efficacy, safety, and side effects, based on that, this data will be submitted to regulatory review. They will evaluate all these parameters. Based on that, they will approve this particular drug for marketing. Once it has entered into market, it will go to the phase 4, which is post marketing. There again, they will monitor the long term safety and effectiveness of the drug in phase 4. So in that way, at any phase of study, if you see, we need to evaluate the amount of drug present in the sample. That is how this clinical sample analysis plays a very important role in drug development process. So this slide tells about what all departments are there in clinical sample analysis. So basically, it starts from protocol preparation team. We have a protocol preparation team who prepares this protocol for each study, wherein they mention how many volunteers should participate in the study, how the study is designed. Once this protocol is written, it will go for ethical committee approval. Once this is approved, then the study will be conducted. There again, there is a separate department who has the volunteer database. Then the volunteers will be called, and clinical team, they will dose these volunteers with the desired test product. So these healthy volunteers will be healthy, or trial participants will be dosed with the test product or the test drug. Once the drug is dosed into them, then we will collect the time points from the patients as per the protocol time points. Like before dosing, they will collect the sample like 0 hour. Then subsequently, after every dosage, then samples will be collected. Example 2 hour, 4 hour, 6 hour, 12 hour, 24 hours, 48 hours, 72 hours, and like that. So this part will be done by the clinical department. Once these samples are collected and stored in the freezer, these samples will be shifted to the bioanalytical team. So by this time, bioanalytical team will be ready with the method development and validation process. Once this is done, I will talk about method development and validation process in detail in the upcoming slides. Once this development and validation is done, the samples will be received into bioanalytical department. The samples will be evaluated against the standard curve to determine the unknown concentration present in the sample. So once this is determined, it will go to the statistical team. They will check again what is the Cmax, Tmax, and other parameters to check whether we got the result or not. If the results are satisfactory, then this regulatory affairs team will come into picture. They will write report, they will draft report, and then they will submit to the regulatory body. So that is how. So every department, there are multiple departments in to conduct a clinical study. So again, I need to mention about the quality assurance team. Before the data goes to subsequent teams, each there is a checkpoint for each operations, wherein quality assurance team will oversee whether it is compliance according to the regulatory requirement or not. So this slide talks about the various departments and their involvement in the clinical study. So that is what. So now let's assume a sample has come to the bioanalytical site. These are the steps which has to be followed to quantify any drug. First, we should develop a method. Once that method is developed, then we should check whether yes, whatever method I have developed is validated. Yes, it is working fine, working to the expectation what we want. Once this is validated, then it will move on to the sample analysis phase. So during method development, based upon the drug we have chosen, we will select the analytical technique what has to be used. Usually for small molecules, we'll go with the high performance liquid chromatography or LCMS method. High performance liquid chromatography is often used for drugs with moderate to high polarity and good solubility in liquid solvents. This HPLC with UV detector is very old technique. Recently, we are using this LCMS. Again, I have most of my experience in handling this LCMS. So here it is preferred for detecting low concentrations of drug and metabolite with high specificity and sensitivity. The main drawback in HPLC is it will not be specific. Based upon this absorption, we will determine the concentrations present in drug. So you will get a lot of interference. Your method will not be specific. You want to quantify drug, but there will be some other components in blood which may interfere in your quantification, and it will tell there are, and you will have high baselines. These are the drawbacks in HPLC. Whereas in LCMS, your quantification will be very accurate. Here it is based upon mass to charge ratio. So even in your samples, you may have the same molecular mass, but getting the same daughter mass, the chances of getting the same daughter mass is very less. So by this means, by selecting this LCMS method, your method will be so selective so that other interferences will not be there in your analysis, so that your baseline also will be very clean when you compare against this HPLC method. Then this gas chromatography is used for volatile compounds. When we are handling volatile compounds, then we go for gas chromatography. Then yes, UV visible spectrophotometry is part of this HPLC. This will be the detector. And currently, I am working in this immunoassays ELISA for drugs where specific antibody-based detection is applicable. Here, currently in our organization, we detect both drug and its antibody by this ELISA method, because mostly it is whatever we are handling here is large molecules which are proteins. So we quantify the protein what we are injecting to volunteers, and then since we are injecting these proteins, sometimes our body may recognize these foreign body as, this will recognize as a foreign body, and it will start generating antibody against it. So because of this antibody, we may lose the efficacy of drug. That is why we detect the drug and its antibody against this drug will be detected to check the efficiency. So for to quantify this drug and its antibody, we use ELISA technique. So the main challenge in method development is, so now as I mentioned earlier, we will receive sample from clinical department. It will be mostly, okay, I'll go to the subsequent. So these many types of samples are there which we may receive from clinical site. It can be either blood or plasma or serum. It can be urine, saliva, feces, skin, cerebrospinal fluid, breath, tissues, hair, or intestinal fluid. Generally, in most common cases, usually we will receive plasma, sometimes serum, and rarely CSF and other metrics we may receive. So based on the metrics whatever we are receiving, we need to quantify the amount of drug present in each of this sample. So the main challenge here is we receive sample. As such, this sample cannot be used for quantification like how we used for other tests. As such, this sample cannot be injected into this test system, whatever we are seeing. Like either it can be an LCMS or a laser technique. These metrics cannot be as such used into the instrument. So it has to be converted into a suitable solvent so that that particular solvent can be used for testing. So there are different extraction techniques and cleanup process to extract the samples from this blood or any other matrix to a proper cleanup sample. Example, we may use protein precipitation method or solid phase extraction or liquid extraction. Again, we will decide this extraction technique based upon the properties of the drug. So then we will optimize the sample volume, solvent systems and extraction conditions to achieve high recovery and minimal matrix interference. Again, this is a very big topic for selection of this extraction technique. So the solid phase extraction technique, it will be more accurate and precise, wherein there will be a cartridge. In cartridge, we will pass this sample in a diluted form into the cartridge. This drug, our drug of interest will bind to the cartridge and it will retain, and other components which is not required will be eluted out of this cartridge. Then we will wash this cartridge with multiple washes so that all other neutral molecules will be eluted out. Then we will change the polarity of the drug, by which the polarity of the drug will be changed and it will be eluted out from the cartridge. So this SPE extraction technique is little more advanced when compared to this protein precipitation and liquid extraction technique. So again, recently also there are a lot of improvements in this solid phase extraction. We get 96 well plate SPE, wherein very limited quantity of sample is enough to quantify the drug. We use 50 microliter or even 100 microliter volumes to quantify the drug present in the sample. So as we discussed, these are the list of clinical samples matrix which we may receive, but the most common matrix we use is plasma or serum. So as we have discussed, so once a method is developed, so we should determine which quantification method you are going to choose, whether it is LCMS based upon the drug, whether it is LCMS or ELISA technique, we should decide. Then we should extract this sample from the matrix to we should convert this into a suitable solvent, and then only we can inject into the instrument. So once this method is developed, like we should take a sample, add some buffer to it, then vortex it, then add an extraction solvent to it. A typical LLE case, if you see, we'll add extraction solvent like TBME or anything. So from this TBME, from this plasma, it will go to this TBME. So this extraction solvent will be extracted, only the supernatant will be taken out. After this supernatant is taken out, then it will be evaporated completely. Then we will add the then we will add a suitable solvent to it. We will reconstitute it. After reconstitution, this sample will be injected into our LCMS system. So this is the complete process. Once this process is developed, we need to validate it. We need to check all these parameters, whatever parameters you are seeing here. All these parameters has to be validated in method validation. Once this is proved, only then we can be sure that yes, whatever concentrations we are determining for samples is accurate. That is why once this process is developed, we should enter into the method validation phase. Again, in method validation, there are a lot of parameters. Let's see one by one each parameter. So first parameter is accuracy and precision. Here in accuracy and precision, we ensure that the analytical method produces results that are close to the true value. This is assessed by comparing the test results with those from a reference or standard method. So we know the nominal concentration. We prepare standards and QCs. For QCs, we know the nominal concentration, whether our method is recovering against the nominal concentration or not, we check. So this is closeness to the true value. Accuracy is the closeness to the true value. The second parameter is precision. Evaluate the reproducibility of the method under different conditions. Example, intraday precision, intraday precision. Those things will be covered in precision. So usually accuracy and precision we evaluate in 6 different runs with standards and QCs at 5 different levels. So here when we talk about standard curve should have at least 6 non-zero data points, and QCs we have at 5 levels. At LLOQC level, which is equivalent to the lowest standard, and ULOQ QC, which is the highest standard point, and in between we add 3 more QCs at LQC, MQC and HQC level. So multiple replicates of QC will be added against this calibration standard, and we will check accuracy and precision of this QC. That is the accuracy and precision experiment. So the next parameter is specificity. So as I mentioned, specificity, next is specificity or selectivity. As I mentioned, this is very important parameter because as I mentioned in HPLC, if you see, it is based upon absorbance. Even our drug of interest will have same absorbance. Similarly, our matrix component, whatever we have extracted, even that matrix component also can have the same absorbance, by which because of that sometimes our baselines will be high and we may not quantify the drug correctly or accurately. That is why the selectivity is very important. So here what we do, we prepare matrix we have matrix without any drug. This will be injected and checked whether anything is getting quantified or not. If nothing is quantified, then which means our method is selective and it is quantifying only the amount of drug. So here basically 3 different levels we check. From different lots we used to check the selectivity. At least 6 different lots we perform. From each of the lot we prepare blank. We prepare at lowest concentration LLOQC and then at HQC level. So here we ensure that the method can accurately measure the drug without interference from other substance in the matrix. So test the method with matrix samples from different individuals to ensure that it works across biological matrix. So whenever we are quantifying, we ensure we check at different individuals. When we run blank, there should not be any interference at our RD of interest, and it is compared against the LLOQ. The limit of deduction, this is the lowest concentration that can be quantified with acceptable accuracy and precision. This is the limit of quantification. Then linearity. This ensures that the method produces the linear response against the range of concentrations expected in the study. Example, from low to high concentration. This is evaluated by preparing a standard calibration curve. So as I mentioned, this linearity and this ANP all together in 6 batches we run calibration standards followed by QCs at 5 different levels, and we check whether the method is working linear or not and whether our QCs are within accuracy and precision or not. That is evaluated in 6 different runs. So whenever we are deciding this calibration range, we consider the dose given during clinical site and we know what is the expected Cmax for those drugs. Suppose if my Cmax for some particular, assume we are dosing 100 mg and the expected Cmax is around you say some 500 nanogram per ml. Then based upon this we construct a calibration curve. So if my Cmax is 500 nanogram per ml, then my calibration, my ULOQ will be 1000 nanogram per ml, at least twice of my Cmax, and my LLOQ will be 500 divided by 32, which is 5 half-life of the Cmax, so that all the time points whatever we are receiving will be within this calibration range. That is the calculation what we usually will have. Our ULOQ will be twice of the Cmax and our LLOQ will be 5 half-life of the Cmax, which means 500 divided by 32. So the next parameter we evaluate is the recovery. This ensures that the method consistently recovers the drug and its metabolite from biological sample after sample preparation steps. Recovery is tested at multiple concentration level. So this recovery is one of the critical parameter or critical challenge what we face during method development, because this is the main challenge or what we face during method development, because we have our test in plasma or serum or whatever metrics it is. Now here we have extraction solvent to it. Now sometimes we may not, this our drug of interest may not come to this extraction solvent, so that is what is called as recovery here. So based upon the drug type, we may have to add suitable buffer to it, so that it gets converted into ionized form and then it may extract into the extraction solvent. So here we check the recovery of the drug, how much drug is present in plasma and how much drug is present in the neat solvent, whatever we are injecting in the instrument. The recovery is very good, then we may achieve very good sensitivity for that method. So usually we'll try to recover the maximum possible, whatever possible from plasma to be extracted into the suitable solvent. So that is about recovery. Here again recovery we evaluate at LQC level, lower level, mid level and high level. And the difference in the recovery should not be more than 15% at each of these level. It should be within 15%. At lower level it should not give some 50% recovery, and higher level it should not give 80% recovery, which means it will definitely affect the linearity. So the variance at all these levels should be within 15%. Only then we can tell that yes, our method is working linear. That is why recovery plays a very important role. Then we should talk about the stability part. So in validation, mainly this accuracy and precision talks about the quantification of drug. That is one part. The next critical part is stability. Here, the main reason why we are establishing stability is whatever conditions we are exposing our study samples, we should check whether the samples are stable in that condition or not. For example, as I mentioned in clinical site, whenever any patient comes or volunteer comes, they are getting dosed with a drug, and frequently at different time points samples are collected from them. Once it is collected, the blood is immediately centrifuged and supernatant is taken, and this plasma is immediately stored in freezer. Then after some time, once all the time points are collected, these samples which are stored in freezer, it is shipped to bioanalytical department. And in bioanalytical department, after once we receive, we take it for analysis and we determine the concentration present in each time point. So these samples usually, if you see, these samples are usually stored in freezer. So during method validation, we should check if we store these samples in -70, whether it remains stable or not. So likewise, there are multiple conditions. We expose this sample, so to all these conditions, whether the sample is stable or not, that has to be evaluated. So here, as I mentioned, so for long-term stability, what we do is we take blank matrix. Here we spike at low and high concentration of drug. We spike it and we store it inside the freezer for the desired storage condition. So this LQC and HQC will be remaining in freezer for that extended period. Assume we are planning to evaluate the stability for 6 months. So after 6 months, the same sample which we have stored at LQC and HQC level will be retrieved. On the day, freshly, we will prepare a calibration curve, and fresh, same way whatever we have prepared 6 months before, same way we will prepare this LQC and HQC again. Along with this sample, the stability sample which is stored in freezer for 6 months will be retrieved and analyzed. So here, when we are evaluating now, we will run the calibration standard, fresh calibration standard, fresh QCs, followed by this stored QCs. Now this stored QC should meet the acceptance of this accuracy and precision. If this QC is passing, which means this molecule is not having any stability issue in freezer. Suppose if it is failing, then we can conclude that yes, this molecule is not stable in this storage condition. Then we may look into some other storage condition, or we may have to add some stabilizing agent to make this drug stable in the condition. So this is the example for long-term stability. Same way, during sample analysis, we will take out the sample and we will keep it in bench top for some time, and then we will analyze it. So whether the sample is stable in bench or not has to be proved. So as I mentioned earlier, so samples will be freshly prepared, kept in bench for some time, then immediately again we will prepare fresh sample and we will evaluate and compare and check the short-term stability. And the next important stability is freeze-thaw stability. Sometimes samples will be subjected to multiple freeze-thaw. So because of that also, the stability of drug may be lost. So this freeze-thaw stability also should be evaluated during method validation. For this, as I explained, we will spike and keep it inside in freezer. Then we will retrieve it. Once it is retrieved, it is considered as one thaw. Same way, multiple times we will keep inside freezer. We will retrieve it. At least for 6 freeze thaw, we will subject the sample, and then for the last freeze thaw, whenever we retrieve, we prepare fresh calibration standard, fresh QCs, and then this freeze thaw subjected sample will be evaluated and verified. So basically, whatever conditions we are subjecting the study samples, we are checking with the known concentration of drug. Here we know this QC sample will have this much concentration. If I am adding 100 nanogram per ml, when I am evaluating after subjecting the samples to the stability condition, it should recover 100 nanogram per ml. So if it is recovering less, which means it is not stable. So that is how we evaluate stability for all of these sample conditions. So the next parameter is matrix effect. So evaluate potential interference from components of the biological matrix, example plasma, proteins, salts, lipids, and determine how it may affect the quantification of drug. Sometimes with different matrix, it may give different interference. That's why whenever we evaluate matrix effect, we evaluate it with different lots or different individuals, so that we make sure even when we conduct study sample analysis, because in study samples we get samples from different volunteers, so that's why we evaluate the effect in matrix effect. So these are the method validation parameters. So once all these parameters has met its acceptance criteria, then we are assured that yes, the method will work fine, and we can determine the concentrations present in unknown samples accurately. Then we will move to the sample analysis phase. So once the development is done, we have a set of process to be done. Once this method is fixed, we are with the same method, we are performing all these parameters, whatever we have seen in validation. Once this method validation is completed, we'll sign off the report and we'll go for the sample analysis. So here in sample analysis, the batch organization will be similar to validation. In validation, we will have standards and QCs. In sample analysis, we will have calibration curves, which is standards, followed by QCs, and then along with QCs, we will have our unknown samples. So basically, all these will be retrieved from the freezer. So before we start sample analysis, we prepare standards and QCs in bulk in different aliquots, and these standards and QCs will be stored in freezer along with the subject samples. So with each analysis, so suppose example, one subject is having some 20 time points. If I want to analyze this 20 time points, I will retrieve this 20 samples along with one set of standards and two sets of QCs. So this calibration curve, the standards, QCs, and this unknown samples will be extracted and it will be injected in the instrument. So based upon the standard response, it will plot a calibration curve, and QCs are there to check whether the calibration curve is constructed properly or not. And from this calibration curve, our unknown sample concentrations are determined based upon the instrument response what is generated for the unknown samples. It is just a back calculated concentration for the unknown samples. So this is our calibration curve. So our x-axis is sample concentration and our y-axis is the instrument response. Whatever you are seeing here, it is area under the curve. So this will be plotted. So for the knowns, we and the response which is plotted, QC also will check whether my standards are, whether my calibration curve is proper or not. So for unknown samples, we will not know the concentration, but we will know the response generated in the instrument. So based upon the response, for example, let's assume this is 10 lakh, my unknown sample, if extrapolate it will be close to 40 nanogram, 40 mg here in this case. So based upon the instrument response, we will determine the concentrations present in the unknown sample. This is how sample analysis works. So as I mentioned, x-axis is the concentrations of, y-axis is the instrument response, and through simple linear equation we calculate the unknown concentration. Okay, so we have seen what are the steps to be followed in bioanalytical department to quantify the drug. Like development we are doing, the of sample analysis is for each sample whatever we have received in bioanalytical department, we have derived the concentrations present in each of the sample. Once this concentration is derived, this concentration data will go for PK analysis. There in PK analysis, they check Cmax, area under the curve, and Tmax mainly. Based on this and other data, they will prepare the report. So it will go for the statistical analysis. So this is the subsequent step after sample analysis, data review and quality control. Here we check whether the data is accurate or not. Once the data is generated, usually it will go for quality control review, quality assurance review. Once the data are found to be accurate and with integrity, then these concentrations will go for PK analysis. Here they, as I mentioned, here they calculate the PK parameters like Cmax, AUC and Tmax. Then suppose if it is a clinical trial, we will just determine the concentrations present in each sample. If it is generic testing, here we check the bioequivalence, wherein we have a reference product and a test product here. Reference product is branded, which is already there in market, and this test product we are checking whether it is behaving similar to the branded drug or not. So for this, usually we will do a crossover study. For example, suppose let's assume we are doing a bioequivalence, means generic drug testing for a drug. Okay, let's assume we are having some 20 subjects as a participant. So for 20 subjects initially, for 10 subjects we will give reference and 10 subjects we will give test. After dosing this, we will collect samples from each individual, and after washout again to whichever subjects we dosed reference, now we will dose test, and for people who are dosed with test, they will be dosed with reference. Like that, for each subject we have this test and reference data. So this data will be evaluated for each subject, how the test and reference has behaved. If the Cmax is same for this test and reference, then based on this data and statistical analysis, it will go for filing. If it is within that acceptance criteria, then regulatory bodies will approve this drug. So that is the statistical analysis part. So then data validation and reporting. Prepare data for submission or internal reporting. So there are separate team to prepare all these reports according to the requirement of regulatory submission. Then followed by regulatory submission. Submit study data for approval. Again, for submission there are a lot of process. If the data are found to be satisfactory and the results, then this data will be submitted to the regulatory. Then post study monitoring. Investigate any irregularities or required follow-up studies. Then conclusion and final report. Conclude study and prepare for submission or publication. Thank you. This is all about analysis of quantification of drug in clinical trials or in BABA studies. If you have any queries, I'm happy to answer. Thank you.
Thank you, Professor. Now the session is open to discussion. Professor, you have mentioned about the different detailed steps involved in the processing sample in the industry, clinical sample in the industry. As a faculty community, from our side, we are training our students. Actually, in our laboratory also, our students are doing extraction work, chromatography relevant works. If our students are, if we train our students in bar with the industry scale, or in what way we have to train our students to directly connect with the industry.
Okay.
And also if you could able to mention about any internship relevant program at your industry or relevant industries whereby an undergraduate and postgraduate can try it. And other participants, if you have any query, kindly post it on the chat box.
That's what Asya asked. What kind of drugs you are handling?
Basically, we uses antibiotics.
Okay.
In our laboratory, we are studying the degradation of antibiotics. So
Okay.
we uses the extraction part as well as the quantification part of antibiotics.
What metrics you are using?
We use column chromatography, wherein we use adsorption chromatography.
Okay. Matrix I'm asking. What is your metrics?
Silica based gel. We
Okay.
haven't defined
Sample.
uses
I'm
any
asking about the
defined
sample. You
samples.
receive.
It is simulated sample only. We haven't
Oh,
received from any
yeah.
samples from biological side. We
Okay.
are using simulated samples.
Okay. Henry, there are a lot of literatures for small molecules. If you see, there are a lot of literatures around, and even in my database, I have close to some 100 molecules. Okay, which we can easily extract. Only few molecules which are endogenous, we may face some challenge. Otherwise, quantification for small molecules like this antibodies and all, it should not be so difficult.
Okay.
I can support whenever you want with respect to quantification or developing a method.
All right.
Yeah. Your which what is your reduction system? You're using HPLC or LCMS?
Yeah, we are using HPLC, and we
Okay.
use here in our laboratory we use only UV detector.
Okay. Currently in industries, that's what I think maybe 10-15 years back they were using UV, but
Okay.
now everybody is upgraded to LCMS.
All right.
Yeah.
And Shiva Nagarajan sir has mentioned that please share the first slide of your presentation, sir. Sir, if you could share the slide and in first slide, yes, sir has some questions.
Yeah, 1 minute.
Professor Sivanagarajan sir, if you have any question, you may mention in the chat also, sir.
It's like this slide or one?
Professor Sivanagaran sir. Okay, he has mentioned and Professor, you have any question?
No, sir. Actually, I need to take a screenshot of the first slide,
Okay.
sir.
All right, Professor.
Okay.
Right.
Yeah, don't worry, I'll share this presentation.
Okay.
And Professor, if you could, Mr. Chandru, sir, if you could tell us in what way we can connect with industry, our students can connect with industry.
Yeah, that's what, as you know, see, I entered into industry through this project or implant training only. Okay, that is the only easiest way you can enter into industry. I did my MTech project in a clinical research organization in Chennai. I did my project for 2-3 months. Immediately after completion of project, they were impressed with my dedication and they gave me job there in that study in that organization. That is the easiest way wherein you can approach an industry. Otherwise, usually it is very difficult unless and until you have some strong reference. It is not that easy to get a job here. Last week, to be frank, last week we had a walk-in interview in Biocon. Okay. Close to 600 people have attended for that walk-in. Morning 8:30, some 300 people were standing in a queue. So the only easiest option is whenever you have project or implant training, try to approach in industry, and that is the only easiest to get a job. Otherwise, it is difficult.
Oh, okay, sir. Thank you. If any more questions we have, we can have here, or else we will wind up the session of the day. Right, sir. If a participant has any question, I'll ping. I will message you, sir. And thank you for your presence and sharing your valuable information. And we'll wind up the session 3 here, and we'll start our session 4 by 7:30 exactly. And you please join 10 minutes before the session starts. And we have posted the feedback link in the chat box. Kindly fill your feedback on this thing. This will be considered as an attendance, and this will not be posted in our WhatsApp group. Chandru sir, thank you. Thank you
Yes.
so much for your time.
Thank you so much, Samal. Thank
Yes,
you.
thank you.
Thank you.
Take care. Participant, you may be connected at this forum. I mean, you may be connected to this link, and you can resume back by 7:30, or you can leave the link and you join the same link by 7:20. Thank you. Thank you for your cooperation. Meet you in the next session.
Thank you. Thank you.
Feedback form for session 3 given in the chat box. All participants kindly fill in the form. Thank you all.
Sir, good evening, sir. Sir, couldn't hear your last instruction. My network was a little bit difficult to hear you. Sir, can you able to hear me? Am I audible, sir?
Ma'am, yes, ma'am.
Yes, ma'am. Yes, ma'am.
Sir, the last something you spoke, but I couldn't hear you due to the technical glitch. My network was somewhat difficult. May I know what you instructed now, sir? Sir, did you spoke something?
Yes, ma'am. Nothing but the feedback link is available in the chat box, ma'am. So please fill the feedback form, ma'am. That was the instruction given, ma'am.
Okay, ma'am,
Yes, ma'am.
I'm not able to see the link also.
Oh, okay, ma'am.
Can you please share the link to my personal WhatsApp, ma'am? If it
Yes,
is
ma'am.
not
Yes,
shared in
ma'am.
the group also, my personal number you can share, please,
Yeah,
if you
ma'am.
don't mind.
Just you message us in the number, ma'am. Given number, ma'am. 9585550185. Kindly
Oh,
give us a query, ma'am.
sure, ma'am. Sure, ma'am. Sorry for the
Thank.
inconvenience. Thank you so
It's
much.
okay,
Thank you.
ma'am.
Thank you.
It's okay, ma'am. Thank you.
Thank you.